ranging from rodent behavioral assays to all the way down to molecular biology and gene targeting technology development, to better understand how changes in chromatin landscape are affected by and contribute to pathologies of addiction and depression. And the Heller Lab applies these techniques to target epigenetic editing and determine how these epigenetic changes are directly related to histone modifications relevant to transcription and all the way up through behavioral changes. Since joining the faculty at Penn, Dr. Heller has received numerous awards, including the Charles Kaufman Foundation New Investigator Award, a Whitehall Foundation grant, a Young Investigator grant from the Brain and Behavioral Research Foundation, and the NIH Director's Pioneer Award. And in exciting news, Dr. Heller just in 2021 received her first NIH R01 grant, which is a big accomplishment. So I'll let Dr. Heller take over, and excited to hear. So please do remember to put your questions in the Q&A, and I'll make sure to ask them at the end of the session.
Thank you so much, Kurt and Christian, for this invitation and the introduction. It's really a pleasure to be here. I'm joining you actually from my home in Philadelphia, as today is a national holiday in the US. And so schools are closed. Hopefully we won't have any little visitors. So let me share my screen with you. Okay. Looks good? Okay, great. So today I'm going to tell you about the project of actually my first graduate student, Marco Carpenter, and he discovered an important role of NR4A1 and its regulation by chromatin bivalency in the context of addiction. So first, I would just like to bring us all to the concept that is on our minds a lot in the lab about addiction, which is the chronic nature of this disease. And in particular, what is sort of especially traumatic and tragic about this disease and unique is that it's really a disease of abstinence. So the symptomology and pathology of cocaine addiction largely begins after drug taking ceases, which is in the form of relapse and withdrawal, or withdrawal symptoms followed by craving and relapse. And this is unique in the sense that if we consider cocaine to be the insult or pathogen that underlies this disease, typically with disease, when you remove the insult, you start to recover, and the body has homeostatic mechanisms and recovery pathways to recover after injury or insult. But in the case of addiction, sadly, this isn't true. And we see in these data from human brain reactivity to cues, visual cues, we see that after just 2 days of abstinence, both drug and neutral cues elicit the same level of activity. But over time of abstinence away from the drug, these drug-associated cues actually increase in salience, and that's underlying the increase in withdrawal symptoms, which ultimately lead to relapse, causing this to be a lifelong disease. And we're curious about how a shorter-term cocaine insult can lead to mechanisms that cause a chronic, even lifelong change in brain structure and function. Our approach to answering this question is to focus on the part of the neuron that is stable. So as you know, most of the parts of the neuron do not last years the way that the disease does, but they turn over in minutes in the case of RNAs and shorter durations, all the RNA and protein in the cell. The chromatin, on the other hand, is stable, and it is consists of the proteins and molecules around which DNA is wrapped in order to make DNA fit into the tiny nucleus. 3 meters of it has to squeeze into a nucleus smaller than a hair, 1/10 the width of a hair. And what we know is that drugs of abuse and stress regulate the chromatin structure and function by modifying histone proteins that make up the chromatin. These modifications are changed across the genome in the context of drug addiction, as well as the enzymes that catalyze and metabolize those modifications. However, it's difficult to causally link chromatin remodeling or histone modifications at one gene to a biological function because these enzymes and modifications occur genome-wide, and we're never really sure if the effect on our gene of interest is direct or indirect. That's one of the things that we're approaching. I also want to just pause to share some of the inspiration for looking at long-lasting changes in the brain, in the sense that Mother Nature has really solved this problem many, many times. In fact, within the cell, there are already cute, elegant mechanisms for making short signals last long. I'll just pause to bring us all back to one of our favorite signaling molecules, CAM kinase. CAM kinase, because it is auto-inhibitory, is unresponsive until calcium floods the cell in the context of an action potential. And although calcium signals are very transient, the response of CAM kinase is to become self-activating, in the sense that a second post-translational modification allows CAM kinase to auto-regulate and stay active much longer than the transient signal. If we take the analogy to the epigenome, there's an example besides histone modifications where we have different types of DNA modifying enzymes working in concert to lay down an initial response to stimuli in the case of a de novo DNA methyltransferase, but then a second class of methyltransferases will maintain that modification long past the action of the initial signal. So we see here that this is something that biological organisms have solved, and I'm looking at some of the mechanisms by which the neuron solves this problem at the level of histone modifications. This is the work, as I mentioned, of Marco Carpenter, now a postdoc at CHOP, and all of the research we'll show today will focus on the nucleus accumbens, a region I don't want to belabor, but we'll mention that it is the recipient of dopaminergic inputs from the VTA and long known a source of epigenetic and transcriptional changes in the context of cocaine. So we use mouse cocaine self-administration in the lab. Again, I think you're largely familiar with this operant conditioning task where animals self-administer cocaine. And we can see that, early on, I started my lab in 2016, have to get the methods to work in-house, and we've successfully done that. The animals take cocaine instead of saline when given that option, and they're able to distinguish between cues associated with a cocaine reward and the absence of a reward. In this project, we collected nucleus accumbens and some other brain regions on one day after the end of a cocaine self-administration session, and again 28 days later. This allowed us to see what others had observed, which is that when we measured RNA transcription through RNA sequencing from this region, we see many genes upregulated, the red dots, or downregulated after one day of cocaine abstinence, but many more gene expression changes occur after 28 days of abstinence. So we recapitulated what had been found in the literature and support our hypothesis that this is a disease during which most changes occur in abstinence. The gene that Marco got interested in is called NR4A1, nuclear receptor 4A1. And he noticed that this transcription factor, also an immediate early gene, was upregulated at one day, but not 28 days of transcription, and that he could replicate this by qPCR in a biological replicate. NR4A1 is an interesting target because it's activity dependent. It's an immediate early gene, and that means it's a transcription factor. We like to focus on transcription factors in our work because their effects tend to amplify since they target many downstream genes. NR4A1 targets its downstreams through the NURE recognition element, which is somewhat degenerate, but fairly well conserved for a TF binding site. It's known to recruit histone modifying enzymes, and fortunately for us, during the course of this project, Dina Walker and Eric Nestler's lab identified NR4A1 in their independent analysis of transcriptional changes occurring across abstinence that associated with what they termed an addiction index. So this was further support for Marco to follow this gene's and try to tell its story. What Marco then interrogated was what downstream targets of NR4A1 relevant to cocaine addiction may be regulated in this context. He profiled many genes, but I'm just going to focus our talk today on this gene, CART-PT. So Marco made an interesting observation. Several downstream targets of NR4A1 were regulated in the NAC across abstinence, as we saw by RNA-seq. But CART-PT was regulated in a time course opposite to NR4A1's activation. So if you remember, at one day of abstinence, NR4A1 was upregulated in expression, and it was back to baseline by 28 days. CART-PT, on the other hand, was not upregulated until 28 days after abstinence. So right away, we have a little bit of a mystery. What signal persisted for these 28 days in the absence of cocaine? Again, this was replicated in a biological cohort by qPCR. So what Marco then wanted to ask was, if we think that NR4A1 is actually part of this regulation of CART-PT, how can we demonstrate that? So we, at this time, went to a cocaine replicate, but instead of self-administration, we used investigator-administered cocaine. This was because at the time, the chip experiments worked better with more material, and this gave us access to that. That is no longer true, which I'll show you toward the end of the talk. So Marco looked at NR4A1 enrichment at CART-PT at these two abstinence points and found, interestingly, that NR4A1 was not enriched at 1 day, but was enriched at 28 days. So now we have a situation where NR4A1 binding to CART-PT is temporarily overlapping with when CART-PT is activated. And both of these occur 28 days after abstinence. NR4A1 is a transcription factor, but it never acts alone to regulate gene expression, as you know. So we were curious about histone modifications that may have been present either early or later in abstinence at the CART-PT gene. What Marco found when he interrogated different modifications was a very interesting pattern. So he first looked at H3K27 trimethylation, which is a mark generally of repression at the gene promoter. What he saw made sense with respect to the pattern of CART-PT expression, which is that at 1 day of abstinence, this repressive modification was enriched at CART-PT, the time point that it was not activated in expression. And later, when CART-PT was activated, this repressive modification was de-enriched. Marco also looked at H3K27 acetylation, generally a mark of active transcription, and this was enriched at CART-PT at both time points. Similarly to H3K4 trimethylation, another probably the most accurate mark of active transcription at the promoter, similarly enriched at CART-PT at both time points. So Marco took this pattern of expression and proposed the following model. In early abstinence, NR4A1 expression is activated, but the protein is not yet bound to the CART-PT gene. In that context, there is enrichment of K27 trimethylation, a repressive modification, simultaneous of these activating modifications. This pattern of expression, where you have both activating and repressive modifications similarly enriched, leads to the CART-PT gene to be poised or ready for activation, but lowly expressed. On the other hand, in late abstinence, NR4A1 expression is no longer activated, but the protein is now bound, perhaps having built up over this abstinence time period. In that context, the methylation mark is de-enriched in the presence of stable enrichment modifications. This allows CART-PT to be activated only in late abstinence. The next question that we asked was whether NR4A1 was directly regulating CART-PT in the nucleus accumbens. So we're measuring this transcription factor at this gene, but we can't really say for sure that this is functional until we perturb the interaction some way directly. So what we did was actually act upstream of CART-PT to design a simple transcriptional activator for NR4A1. We reason that if we remove the cocaine and focus solely on the up or down regulation of NR4A1, we could confirm that it is sufficient for activation of CART-PT. So this is using CRISPR technology, which in our context, we use a CRISPR tool that does not act as a nuclease. This is the dead Cas9 molecule in which we can fuse dCas9 to a different effector. In our case, we're not interested in nuclease activity, but rather activating a gene with VP64, repressing a gene with the CRAB domain, or in another context, using a tag, an affinity tag, to prove the specific targeting of our Cas9. So Marco built these tools and designed guide RNAs, which would recruit this dCas9 fusion to NR4A1. What you can see here is the initial guide design. This is a technique that our lab uses very often, and I'd like to give you just a little bit of detail today so that in your experiments, you can have a little methodology, and also please always feel free to reach out to us. We tend to design guides that span roughly plus or minus 250 base pairs from the transcription start site, with the expectation that guides targeting downstream will be repressive as they can block transcription off the promoter, and guides upstream may be able to be transactivators or repressors. We express these tools in the nucleus accumbens using a combination of stereotaxic surgery and neuronal specific promoters. In this case, it's H-human synapsin promoter that's driving expression of the Cas9. And what you can see here targeting on the nucleus accumbens is that indeed we're able to express our Cas9, which this is using a Cas9 antibody actually, co-localizing with neuronal expression and never in non-neurons. The next part of the project is to screen these guides for efficacy. I again want to point out the use of control guides in these types of experiments. Also, it's very useful to have both an activator and oppressor effector domain, because then you can set your expectations for bidirectional regulation. So in this case, this is in the nucleus accumbens. You can see that when our NR4A1 guide, in this case, we've narrowed in on this guide as the most effective in some neuro2A cell validation experiments. Now in nucleus accumbens, we can test that compared to this non-targeting guide, which doesn't target any sequence in the mouse genome, we're able to activate NR4A1 expression in the nucleus accumbens or repress it, depending on if we have VP64 or CRAB DCAS9 on board. You can see that there's, we're going to look at NR4A1 expression if we use a non-related guide, CDK5. You can see that there's no activation of NR4A1. And similarly, we always like to see that we don't get repression when we use the activator and vice versa. So getting back to our actual question at hand, is NR4A1 sufficient to activate expression of CART-PT? And for this, Marco injected the DCAS9 VP64 and the NR4A1 guides, which we'll now just refer to as CRISPR-A for activation, into the nucleus accumbens. And what you can see is that CART-PT is activated in the context of NR4A1 activation, which means that NR4A1 is sufficient to activate CART-PT. Similarly, if we repress expression of NR4A1, we see a repression of CART-PT expression as well. The time course here is relevant. This is about 1 week after injection of the DCAS9 tools. Now we were curious to see if NR4A1 is sufficient to regulate CART-PT expression, is it doing so in the same mechanism that we observed in the cocaine context? And Marco found, indeed, that when NR4A1 is present and CART-PT is activated, H3K27 tri, that repressive modification, is similarly reduced at CART-PT in the context of, in this case, an activation of acetylation or enrichment of acetylation, and no change in K4 trimethylation. So although the pattern doesn't match exactly what we saw in cocaine, we can say that in a similar way, CART-PT before NR4A1 expression is sitting in a somewhat repressed or lowly activated poised state where there are similar levels of repressive and active histone modifications. However, once NR4A1 is expressed, then we find that the repressive modifications are de-enriched in the context of activation, increasing and in some cases being retained. So this gene is going from poised to activated. Importantly, in all of our experiments, the targets that we choose have to be important for the mouse at the system level. We'd like to see that these perturbations are relevant to cocaine-induced behavior. Otherwise, we may not choose to really study deeply the mechanism there. In this case, Marco used the CRISPR tools to test for regulation of cocaine reward in the context of overexpression of, or activation of NR4A1 expression. This is the cocaine conditioned place preference paradigm, where animals are trained to, animals are first allowed to explore an open two-sided chamber, and the amount of time they spend in each chamber is recorded. You can see here that at the beginning of the experiment, the animals spend the same time in each, it's actually shown as no preference in each compartment. Forgive me, that was a little confusing. After pretest, we then expose the animals to cocaine and one side of the chamber in the morning, or saline and the other side of the chamber in the afternoon. And then we examine their affinity to spend time in one or the other compartment as a measure of how well they prefer/remember the cocaine-associated exploration. What we saw was that in the dose of 10 milligram per kilogram training dose, we see that animals do prefer the cocaine paired chamber to a greater extent after training. However, when animals were given NR4A1 expressional activation, this reward decreased. Similarly, at a higher dose of 20 mg per kg, you can see that the animals prefer cocaine similarly, and that behavior is attenuated when NR4A1 is activated. Interestingly, CART-PT expression was also activated in this context. There's two things going on here, which is important to point out. We somewhat expect CART-PT to be activated because cocaine is on board and NR4A1 activation is on board. And I've already shown you that both of those things activate CART-PT expression. What was unexpected is that NR4A1 wasn't activated. And we think this might be because of a ceiling effect of cocaine, which cannot be increased with the CRISPR tool in this context. We found similarly that bidirectional behavior was elicited by this approach. So when we repress NR4A1 expression, and we're able to augment cocaine conditioned place preference. Marco then went on to look at the effect of NR4A1 on self-administration, as this is a more robust and comprehensive paradigm for measuring cocaine reward behavior in mice. And as you know, it's the paradigm in which we identified NR4A1 in the first place. What you can see here is that when the NR4A1 CRISPR activator was administered before the initiation of cocaine self-administration, there was no effect. So animals acquire self-administration behavior and take roughly the same amount of cocaine in the presence or absence of NR4A1 activation. On the other hand, at 1 day of seeking, there's no phenotype. Marco then thought that this activation time point is not the most likely to be effective, since we know that NR4A1 is naturally activated by cocaine early after abstinence and is likely activated throughout the paradigm. So we may not be able to elicit much of a response from animals by adding more NR4A1 at this time point. On the other hand, Marco also showed that at 28 days, NR4A1 repression was back to baseline. So he thought, let me treat the animals with the NR4A1 activation at this late abstinence time point when NR4A1 should be down. And in this context, again, there's no treatment during acquisition, so there's no phenotype, there's no batch effect. But on the seeking experiment, after late abstinence activation of NR4A1, you can see that there's a suppression of seeking behavior. This was one of the more exciting results I've gotten in my career. Most of the molecules that I've targeted increase cocaine reward behavior, which is very good for developing biology, but not great for thinking about therapeutics. So at this point, we felt that we had a model that actually Marco brought this concept to the lab. And I'm going to introduce here how we're interpreting the function of NR4A1. So as I showed you in the beginning, after 6 months of abstinence, cocaine-related cues tend to increase in their elicitation of a neuronal response in human addicted patients. However, there's something promising as well. If these patients are followed out for a year, those drug-addicted cues, if no relapse occurs, actually can weaken in their ability to elicit response. This means that the brain has some homeostatic mechanisms endogenous that are able to reverse the effects of cocaine if users can stay abstinent long enough, and that's a tall order, but a promising concept. Marco then started to rethink what role NR4A1 and its downstream targets are playing in the context of cocaine abstinence. We know that maladaptive changes are occurring in the beginning of abstinence, and these changes are what lead to that increase in cue intensity in those first 6 months. However, at the same time, homeostatic changes are working to reverse the deleterious effects of chronic cocaine exposure. And over time, it may be possible for these two to come into balance, the homeostatic changes actually to take over and for patients to recover. The idea is that when homeostatic changes are effective, those cue and drug sensitivity goes down and the threshold for a relapse is much higher, which is really what the goal is for patients. It seems like if we can get them over the literal hump of withdrawal symptoms, they may end up in a neurological space of endogenous healing mechanisms, which is true for most diseases. So we started to think, is NR4A1 a master regulator of these types of abstinence-induced homeostasis? And the second question is, if that's true, could NR4A1 be a good therapeutic target? I really have to credit Marco for all of this. It's not typically where I would imagine my work going. And we've started to make little headway into this. And I would like to share the beginnings of the story with you today. So Marco thought, I think we should pursue this. The fact is, as you well know in this audience, there's no therapy for cocaine substance use disorder. There's nothing we can offer patients besides behavioral treatments, which suffer from incomplete compliance. As you know, there's stigma around therapeutic and neurobehavioral treatments. They're often difficult to get to, and all patients deserve an option of a variety of treatments. The fact is that all cocaine substance use patients will relapse. There's no really getting around that. And so there's a desperate need for additional medications and treatments for this population. Importantly, it's also important to mention, this is the slide, cocaine abuse is stable. We see epidemics occurring in our population. We've just gotten over a traumatic epidemic, or still in, of opiate use disorder. Personally, I think we're going to have some issues coming up with marijuana use disorder as legalization continues to flow across the country, as it should, but we have to be aware of these epidemics. On the other hand, cocaine use is stable. There's always millions and millions of people suffering from this disease. The population never wanes, and that's something we also have to be aware of. In fact, the number of overdose deaths increased for cocaine in the last decade. So we can't ignore this, even when it's not, the epidemic of the day. So Marco, remember, was able to show that a CRISPR tool given during abstinence was able to attenuate drug-seeking behavior. This result was key for us because, as you know, drug-addicted patients, they come to the clinic during abstinence, tend to show up, in unideal circumstances after an overdose at hospital, and through that experience, end up hopefully in rehab treatment. So we can't capture these patients any earlier, really, than when they're in the throes of relapse and withdrawal. So finding any perturbation, as many others have in the field, that can decrease drug seeking after both a chronic taking and long abstinence was very exciting. Also exciting was that small molecule activators of NR4A1 were already extant. So the first molecule that Marco tried is actually a small molecule from Sigma called cytosporin B, naturally occurring, which we found can activate NR4 expression in the NAC when administered IP. And Marco then performed a few different treatment strategies just to get us started with using small molecules to regulate gene expression and behavior. And what you can see here is that with 3 different treatment strategies, number of doses over particular days, NR4A1 was activated. So this is a very robust way to activate NR4A1 in the NAC, although this is bulk tissue, so I can't say that this is necessarily exclusively neuronal. Downstream of NR4A1, we also see CART-PT expression when we use SynB, although it's not as consistent in this set of experiments. Marco then was curious to see, as we're interested in epigenetics, if this drug was also causing NR4A1 or CART-P regulation in the same model that I had shown before with the loss of repressive modifications. However, we don't think that this is really recapitulating that mechanism. We do see a similar enrichment of H3K27 trimethylation. However, that enrichment doesn't seem to track with the activation of CART-PT quite directly. And we don't necessarily see changes or enrichment in K4 trimethylation at CART-PT in this context. So we think that there's a different downstream mechanism by which NR4A1 is activating CART-PT, or perhaps it's not a direct activator in the context of this drug. Of course, the time course is very different. And as I'll show you in a few slides, using a drug is not as simple as I thought it was. So then Marco went on to test SynB in the context of cocaine condition place preference. The fact is, for drug development, if we can get a phenotype, we may worry about the exact mechanism in a different order than we would as basic biologists. What we can see really promisingly is that similar to the NR4A1 CRISPR activation, SynB can also lead to attenuated cocaine condition place preference. And in that context, both NR4A1 and CART-PT expression was activated. This effect was this paradigm we repeated, where we wanted to see if SynB could affect their reward preference, their place preference, even if we treated after training, trying to get a slightly approaching that abstinence time point. Although, of course, this is not considered any type of long abstinence or chronic taking. Fortunately, though, we were able to attenuate their cocaine condition place preference even when we treated with SynB after the animals had received training, which to our mind means that SynB is able to activate NR4A1 somewhat above the ceiling we observed in those CRISPR experiments. And perhaps that is the reason that we were able to see enriched NR4A1 after cocaine treatment with the SynB drug. At this point, Marco alerted me to a new initiative from NIDA for a small one-year grant to characterize novel targets. Again, if Marco wasn't motivated, I probably wouldn't have applied for this, but we did. And it was really meant for, and there are coming initiatives, which I will also show you, it's meant for basic scientists that really don't think about drug development. And part of, this is like a little PSA for NIH, but I'm going to do it. And the part of the reason, which, you know, I'm reluctant to go into this space for very many reasons, mostly my relative ignorance of it. But the truth is, the pharmaceutical industry isn't touching drug addiction. There's minimal investment in this space. And the pharmaceutical industry likes other people to fail at finding good targets before they, you know, put their eggs in any baskets, before they make risky investments. So actually, by basic scientists testing targets and using NIH initiatives to do so, even if we fail or succeed, for pharma, that's de-risking the field. So I've gotten somewhat convinced by really great scientists and program officers at NIDA that this is part of my responsibility as a drug abuse researcher. I don't know yet if I'm convinced, but I think it's an important thing for all of us to think about. If pharma isn't going to test new targets, who's going to do it? So we developed a program in order we were going to test actually two small molecules, both the SynB that I mentioned and a novel class of molecules from Dr. Stephen Safe at Texas of TAMU that are really specific activators of NR4A1 in the context of cancer, primarily breast cancer, in which NR4A1 is implicated. So we had some nice small molecules to target. I won't show you any data, but I will tell you that what mostly came out of this grant is that both of our small molecules are very, very rapidly metabolized. So what would have been me showing you mass spec of metabolites is me not showing you any data because you can't find any. It does SynB have a half-life that approximates some other neuropsychiatric drugs, but we still have our work cut out for us. Fortunately, there's another RFA similar in scope for those of you that are intrigued by what I've told you today, came out from NIDA and is due in February. So we're really going to continue this work with our collaborators at Penn. Now I will share the last little bit of the story, which is what is on our mind after discovering sort of the relevance of NR4A1 to behavior in the context of cocaine treatment. Which cell type? So this is the question for all of us studying the brain in the last 5 to 10 years. And in particular, in the nucleus accumbens, I think we get obsessed with this question because we have two cell types that we find working in opposing ways. And we find this on a molecular level. I actually have been noticing, I think often the DRD2 or A2A population, they're somewhat silent on the matter of gene expression, and you see more changes in DRD1 cells. But either way, looking in bulk, we are losing signal, and it's very difficult for us to tell the true function of our molecules. Maybe NR4A1 works in every cell in the region, but we've got to start to get at this question. So the approach that we take as a first pass is using Cre-dependent CRISPR NR4A1 activation. It just means that we have a Cre-responsive element upstream of those DCAS9 fusions that I showed you. Those tools are all built by Rachel Neave. And we can then express activation of NR4A1 CRISPR tools in just one or the other pathway. In that context, we can then measure all of the things that I showed you before: gene expression, histone modifications, and behavior. And what about those histone modifications in specific cell types? Well, first we've got to isolate them. So now I'm going to talk a little bit about the method we've been working on to isolate these two cell types in such a way that we can get not just gene expression, but also chromatin profiles, which is not trivial because it's not a lot of material. So we are using a combination of strategies. Intact is the first strategy. This comes from Alyssa Moe and Jeremy Nathans a few years ago. And in these intact animals, you have either A2A cells expressing Cre-recombinase or DRD1 cells expressing Cre-recombinase. And then we can inject in them our Cre-dependent, that one cell line expresses Cre. The other mouse line expresses the SUN1 GFP fusion. This is what is primarily the functional element of the intact method. That SUN1 GFP fusion labels nuclei. So now you have an opportunity to use Cre to label nuclei only in your cell type of interest. And then you can immunopurify those GFP labeled nuclei. SUN1 is a nuclear transmembrane receptor, in order to isolate a pure population of A2A cells. Many of you have, I'm sure, know about isolating these cell types by FACS, which is also a useful method, but I'll show you in a few slides why we want to use affinity purification in this context. We can also, using these animals, express Cre-dependent guides, which will then only be functional in one or the other cell type of interest. So with this amazing intact mouse, we can purify and regulate specific subtypes of neurons. This is data showing as validation by mRNA expression of this is showing NR4A1 and CART-PT expression in the A2A cells isolated using the intact method. What we do here is compare the enrichment of the NR4A1 transcript in the IP'd population of neurons from the whole rest of the mix. And what we see is that the NR4A1 is specifically enriched in the IP'd A2A cells and not in the rest, which is indicating to us that this transcript is enriched in A2A neurons relative to the rest of the nucleus accumbens cells. In addition, CART-PT is enriched in that cell type as well. In the context, sorry, I'm saying this in a weird way and I apologize. In this experiment, when we use NR4A1 guides, specifically in A2A neurons, we can activate expression only in the purified neurons and not in the rest of the cells. And downstream of that, CART-PT follows. I was confusing this with validation data, which looks very similar. We then, knowing that we can activate Neuro2A expression specifically in the A2A population of cells, we then asked if this was sufficient to regulate that attenuation of cocaine place preference. And we find that it not only attenuated, it actually reversed in this experiment the mice preference for the cocaine paired chamber. This is a preliminary data. This is unpublished and is an N of 1 experiment with an N of 3 to 5 mice per condition. But the phenotype is striking. And in some ways, we worry that we may cause aversion targeting just the A2A cells. So that's something important to consider. I also look at this data and think to myself, our signal is increasing. So often when we go more specific in the lab, we're worried we're going to lose our effect. But I'm actually starting to be convinced that in the NAC, when you go cell type specific, you always get a better signal. And that just proves the point that we're actually muddying our effect or signal when we affect the entire region. Always, it's important to show that there's no locomotor effect, and that's what's shown here by the total distance traveled. So then Marco asks the same question. Was the mechanism of de-repression of K27 tri the same when we look in a cell type specific way? Although here we have to pause, because at the time of asking this question, there was no way to purify specific MSN cell types for chromatin profiling at this level. That is where this team of Delaney Fisher, current graduate student, Marco and Shuo Zhang, a current bioinformatics postdoc, developed what we eventually termed Icarus. The Icarus method brings together the intact method for isolating the neurons with a novel, relatively novel method, cut and run, for chromatin profiling. So why cut and run? First reason is that cut and run from Steve Henikoff's lab, if you're not yet aware, perhaps you are, has a much better signal to noise for the chromatin profile after sequencing, as well as the fact that it's an on-bead method. So that means when you perform cut and run on nuclei isolated from any cell type, the nuclei are immobilized on bead during the biochemistry to isolate the bound DNA. Because we isolate intact tagged nuclei on bead, I presume that we could fuse these two methods with relative ease. And the PI always thinks things are with relative ease. And it took us a while, but now we've got it working. So what you can see here is validation of our isolation of A2A and D1 enriched cell populations by looking at the SUN1 GFP and this is the SUN1 GFP and the A2A cell. You can see the nuclei are labeled by SUN1 GFP, or in the opposite genotype, DRD1 activates SUN1 GFP in the DRD1 population. We then isolate these nuclei using an anti-GFP antibody on bead, and we have now our GFP positive nuclei isolated and immobilized before we perform cut and run. These are some preliminary data. This is at the step after isolation of the nuclei, and then after affinity purification and immobilization. This was the data I thought I was showing you before, forgive me. So these are the validations of the specificity of our immunopurification approach. And we're looking at the immunopurified population versus the supernatant. And it's important, what our expectations are. So our expectation is that when we isolate DRD1 cells, DRD1 is enriched in our IP relative to our supernatant. And that's essentially what you're seeing. And so similarly, we expect, here's A2A to be enriched in the IP relative to the supernatant, because that tells us that our population is being specified for that gene expression. We also expect to see the DRD1, the A2A de-enriched in the supernatant when we isolate A2A cells. So similarly, we can show the opposite order. I apologize if I'm pointing in a confusing way. The key thing for us, after validating that A2A is enriched in the IP of A2A isolated cells, and DRD1 is enriched in the IP of DRD1 isolated cells, and the opposite are enriched in the opposite supernatant, we can similarly confirm that NR4A1 was, NR4A1 was, I can't see it here now. I'm getting confused. It seems like on this experiment, DRD1 is not enriched in the supernatant at A2A cells, and NR4A1 is lost in the supernatant of the DRD1 cells. CART-PT, on the other hand, is enriched in the supernatant, is enriched in the IP of the A2A cells and not enriched in the IP of the DRD1 cells. So that's giving us some specificity, although I think that there's some inconsistency in the data I just showed you. And I guess this is what happens when I show new data. So assuming that we have specific enrichment, the next step was to profile K4 trimethylation in these specific subtypes. This is the work that the group has been doing. And what I'm showing you here are validation data of K4 trimethylation sequencing profiles and K27 methylation an activating and a repressive modification that were relevant to the CART-PT activation that I showed earlier. H3K27 tri was the modification that was de-enriched at CART-PT in the context of cocaine addiction. What you can see here is that when we look across, we can see that we're able to validate replicates of DRD1 and A2A, 2 replicates per condition versus bulk NAC for K4 trimethylation profiling, and that these replicates show greater correlation with one another than either do with an IgG used for the cut and run experiment. Importantly, buried in this data is a very in-depth profiling of different antibodies for these applications. So not all antibodies will show this level of specificity between replicates relative to the IgG control. We also used bulk NAC chip sequencing data to confirm the specificity of our chromatin profiles. And that was an important control to know that our cut and run profiles in each cell type somewhat overlapped with the bulk chip sequencing profiling available in the literature. What we see here is the relative enrichment of the K4 trimethylation genome-wide in the 2 cell types. What you can see first is that the K4 trimethylation activation mark is overall less enriched, meaning the extent of the in size of the peak enrichment is less at every gene profiled in the 2 cell types. Pardon me, this is K27 tri. We can also see by heat map that we have the expected enrichment of K4 trimethylation as a very narrow peak across expressed genes in this specific cell type. Importantly, we're using gene expression data in some cases, as I'll show you, from published cell type specific transcriptome sequencing studies. So these validation studies are hopefully giving some people optimism that they will be able to profile chromatin genome-wide and specific cell subtypes. It's nice to show tracks because it makes everybody feel more confident, including us. And at this gene, CART-PT, when we're becoming quickly our favorite, you can see that there is some difference in the profile where this is quantifiably A2A expression of A2A enrichment of K4 trimethylation is greater at CART, is greater than in DRD1 cells, which is somewhat consistent with the RNA qPCR data I show you here, where CART-PT is specifically enriched in the A2A supernatant, but not A2A IP, but not in the DRD1 IP. So that is consistent with our seeing greater enrichment of the activating modification in the A2A cells. How this profile changes in the context of cocaine is the next question. And I will leave you with the model that we are currently investigating, which is the concept of chromatin bivalency. It was in the title, so it has to be in the end of the talk too. And it's really a nod to where we're going next. A lot of the story I told you today was about these 2 modifications, K4 trimethylation and K27 trimethylation. These 2 modifications have told the story before in the context of differentiation and cell division during development. In particular, the presence of these 2 modifications at one gene that's slowly expressed has been called a bivalent gene, in that it has both activating and repressive modifications simultaneously. When a gene is only enriched in K27 tri, that gene is typically silenced. In this case, the sails of the boat are down. It can't go anywhere even when the wind blows. On the other hand, a bivalent gene, the sails are up, but there's no wind. The gene is ready for activation once there is a signal. We think that this is what CART-PT enrichment of K27 and K4 tri is doing at that 1-day time point. The cocaine abstinence is the wind, and we see K27 removed from CART-PT, gene K4 tri retained, and gene expression taking off in late abstinence. We're now investigating if these bivalent genes exist in the context of the nucleus accumbens. And for that, we're using specific profiling in specific cell types. Since bivalency is somewhat laid down during differentiation and development, it's important to study it in a cell type or single cell specific way. What we're seeing so far, interestingly, is that when we use gene expression as an anchor for our expectation of bivalency, we can see that as for genes that have primarily K4 trimethylation, you have the greatest level of gene expression. For genes that have primarily K27 trimethylation, you have lower gene expression. And genes that have both modifications are intermediately expressed in specific cell types in the nucleus accumbens. This is an analysis of A2A cell expression. With that, I will just promote myself by saying that I've done another recorded talk for Worldwide Neuro, where I talked about a paper that we just published in Neuron. And so if you knew about that and were hoping I was going to talk about that, please just look at the recorded seminar. There's a lot of great stuff in there as well. And with that, I would love to thank my lab and my collaborators. This is us going from being virtual to being in person. Thank goodness. It's an incredible team, and especially wanting to point out Marco, Delaney, and Schwo for their efforts, and our various collaborators. All of the funding that was mentioned by Kurt, thank you, and to you for your attention.
Great, thank you so much. And now, Kurt, why don't you start the Q&A?
Great. So if people have questions, remember to put them in the Q&A. I guess I'll start with one that I had, which is, I guess I was kind of surprised to see the function of CART, which I think of as a peptide, like a neuropeptide that's released as a sort of transcriptional regulator. So I was wondering if you think that CART in the accumbens is sort of having a dual role, both functioning in these neurons as a neuropeptide that's being released and altering downstream functions and as a transcriptional regulator at the same time, or?
I mean, every day I'm looking for the receptor.
Yeah.
Checking the literature. No one can tell me about this peptide. So they discovered it because it seems to be upregulated when you treat animals with amphetamine, right? And that was like the end of the story. I mean, other people have tried expressing it, and I regret I can't do justice to a literature that may exist, because as far as I can tell, it's exactly as you described. It's known to be present at a protein level after exposure to psychostimulants, but it's unclear what it does. I think that that can be a very key part of the project. And in fact, we've recently with Julia Winter, our new postdoc, she's designing guides for CART-PT because we want to see if we can upregulate that. You can also inject the peptide, apparently.
Yeah.
So it might be homeostatic, though. There is a little bit of suggestion in the literature that it reverses dopaminergic excitability. So it goes with the story, but it's a very mysterious peptide.
Yeah, it's interesting too, because I know it's also like super enriched in like the glutamatergic inputs. Like I think about like the PVT, it's like one of like the densest expressed peptides in like PVT that like in those neurons that go to accumbens and like context induced.
I mean, the two subtypes receive glutamatergic input as well, not just dopaminergic. So you're right, we could be homing in on a signaling downstream of glutamate. That's a good point. That's a really good point.
Yeah, I think the peptide aspect is just so cool. Okay, so then we have a question here from Ankush Chakraborty. They're wondering about if there's any way that extracellular vesicles could affect CART-PT or NR4A1 in the accumbens, and if you have any idea about potential mechanisms.
So if I'm reading the context correctly, these extracellular vesicles could be coming from glial populations. I mean, I'm hearing and reading a lot of exciting news about microglia releasing vesicles in the context of neuroimmune reactivity. And of course, astrocytes, we know, release neurotransmitters. So I don't know. I certainly haven't looked, but I think that you're absolutely right. It is an important place to look, especially when we think about epigenetic regulation, because vesicles are being found to hold small RNAs. Colin Koenig is at Penn now, and he studies epididymal vesicles for transmitting microbes to the sperm. So different context, but similarly difficult to purify. So I have some good people to work with to answer that question. Thank you.
And we have another question that is wondering about the gene chromatin bivalency. And the question is wondering if you're looking at both a cell type specific way and allele specific way. Otherwise, the questioner thinks that it could just simply be like a mixed population that's driving the effect.
Exactly. So we have to take care when we define things as bivalent. And there's a reason that the first 50 slides didn't use that term, because I'm cautious when using it. If we want to consider things as truly bivalent, there's two things missing from my analysis. Even if it was the specific population, for bivalency to occur, I would first purify the K4 trimethylated histones, and then from that population, purify the K27 modified histones. And in that way, with this sequential profiling, we can say for sure that the two histones are on the same nucleosome population. That work is being carried out by Delaney Fisher right now, and we have some interesting results that suggest NR4A1 is truly bivalent when we consider it from a nucleosome-centric view. However, that still doesn't tell us if those nucleosomes are all in the same cells. Maybe some cells have bivalent nucleosome and monovalent nucleosome. Sorry, not same cell, but same population. The benefit of the sequential chip is the fact that there's only one genome per cell. So that helps us because at least we know if we're seeing the mods on the same chromatin, that chromatin has to be coming from the same cell, right? Which is helpful, but it doesn't tell us the cell type. What we have not yet been able to do, and I don't know anyone that can, is sequential chip on a single population of neuronal nuclei. The material right now is just really, really low. I wonder if it will be something solved actually by imaging, perhaps with something like, a peptide-based method to find those and label those particular labeled histones. But at this point with biochemistry, it's very difficult to solve. So we do the chromatin profiling in a cell type specific way, and then the sequential chip in a bulk way. And we're hoping that together we can suss out true bivalency from what we'll just call combinatorial chromatin. But key question.
So if I may, I was wondering to which extent you think that what you see is specific to cocaine, or would similar things happen with other drugs, or even with a sort of a generic way of looking at addiction by stimulating strongly the dopamine neurons in the VTA, which leads to similar behavioral changes. And so the question would be, would you have similar changes in the assays you did look at?
Yeah, so it's a great question. We haven't tried other drugs of abuse. So I can say I personally don't know that. We do have sucrose self-administration data, and we don't see the same pattern of gene expression for NR4A1 and CART-PT in that context. Actually, that was one of the ways we selected NR4A1, was that it was somewhat uniquely regulated in the cocaine context and its downstream targets. I do have my Schedule I license. So we do study opiates. Actually Marco published a paper on opiates, as well as really cool work with marijuana-based what's the word? These chambers where they do the vapor chambers, with Mariella De Biasi. So I have tissue treated with both of these conditions. And I absolutely agree. I think especially psychostimulant versus opiate may be a very interesting comparison given the locus of the effect, but also this business about CART-PT, which doesn't seem to be really a key regulator in the opiate context, as far as I know. We do have the profiling from other brain regions as well. So there's a few things that we're pulling in, but it's a key direction.
Good. Kurt, you have some more questions, or are we? Yeah.
I think that was going to be my last question. So you
good.
Had the same.
Fantastic. So I guess we're exactly at the hour. So thanks again, Liz, for a very interesting and stimulating talk.
Thanks,
and
everyone.
all I have to do now is to remind you that next Thursday, we're going to have another talk by Leon Coleman from UNC. So please tune in next Thursday, 6 p.m. in Europe, noon at the East Coast, and 9 a.m. at the Pacific West Coast. Okay, so thank you so much and enjoy the rest of the day. Thanks, Liz, for having been here. Thank you. Bye-bye.